Sure. Hello, everyone. My name is Jiang. It is a great opportunity for me to be here and share with you my project titled Bayes Miles. That is a Bayesian segmentation model for longitudinal epidemiological studies. So I think the previous speaker have given us a pretty thorough review of the background information of COVID-19, and also the last speaker just mentioned several statistics about the COVID-19 in the United States. Well, we see that during this very hard time, our local governments have introduced different public health interventions to prevent the wide spreading of this new coronavirus. Well, since we're talking about the interventions, actually different strategies could either slow down or speed up the disease spreading. So as all of these different types of intervention measures listed here, only some of them could bring actual influence to the disease spreading. So we are wondering what are those relatively more important interventions, and when did those interventions bring some actual changes to the COVID-19 spreading? To answer this question, we choose to study the temporal changes in the observed longitudinal COVID-19 data. So our model, Bayes Miles, aims at detecting the transition points that partition the observed temporal data into different stages or segments. And the transitioning from one segment to the next could be due to the fact of intervention effects if we can match the transition time with the time when the interventions is announced. And if the time can match, we can further evaluate the effectiveness of interventions by quantifying the transmissibility in a certain stage. Well, on the other hand, the transition across stages could also be due to events such as virus mutation. But for such type of events, it is usually hard to determine the exact time when it first appeared. Now you may wonder, how should we quantify the transmissibility of the disease? Because our ultimate goal is to quantify how effective were those important interventions. So to answer this question, we can use the SIR model. Again, SIR model is used to describe the flow of individuals through three mutually exclusive compartments. Those are susceptible, infected, and recovered. And here are two important parameters in the traditional SIR model. First is transmission rate. And in Bayes Miles, we assume that this quantity will stay as a constant within the same stage, but it changes across different stages. And the second parameter is recovery rate. And according to several recent studies of COVID-19, it is typical to assume that the recovery rate of COVID-19 stay a constant over time. To quantify the time-varying disease transmissibility, people typically use the ratio of beta over gamma, which is referred as the reproduction number. So before we dive into the details of Bayes Miles, I would like to first show some result when you apply it to the real data. So basically, Bayes Miles will help us to answer the two questions, when and how, regarding those important interventions. So when applied to the data we observed in Texas, our model detected 5 transition points as marked by the vertical lines here in this figure. And when we're taking a closer look at these dates, for example, the first change point happened on March 28th. And on the same day, we noticed that CDC issued the 14-day domestic travel advisory. The second change point happened on April 9th. And on the same day, our Texas governor extended the state disaster declaration for another 13 days. So presumably, both of these two interventions should help us to control the spreading of COVID-19. And in Bayes Miles, we also estimate the reproduction number for each stage, as shown by the red dashed line here. And as you can see, when we are transferring from the first to the second segment, you see this downgoing trend for the reproduction number. And it is also true when we're transferring from the second to the third segment. So these results reflect that our model can provide our estimation that can reasonably reflect our understanding about these intervention effects. So we will go back to this figure later in my slides. And our base models can also include predictions based on the SIR model. So here we consider a 2-week prediction of the new daily cases given by 2 different strategies. And in this page, I would like to emphasize it is important to consider the disease dynamics when you're making prediction. So so the proposed method makes the prediction based on the information from the most recent segment. But another alternative method, which we referred as the full data SR model, will fit a single SR model based on all your observed data. So when we're trying to compare the prediction results, as shown here, our predictions marked by the red triangle here matched pretty well with the real data as shown by the black dots. But the full data SIR, the prediction given by the full data SIR model tend to overestimate the daily confirmed case in this situation. So that means if you ignore the COVID-19 dynamics, there could be some issue with your prediction accuracy. Next, let's start to see some details of our model. In general, we detect those transition points that partition the data into capital K of distinct segments, where each segment is characterized by a segment-specific reproduction number. So this value Rk will be a constant within the same stage, but it is changing across different segments. That will reflect the transmission dynamics of COVID-19. And from a statistical point of view, we treat those transition points as the change points. So next, let's see how we define the change points in our model. Well, first of all, our change point detection is based on the daily actively infectious count data, which is essentially the compartment of infected count, IT, in the SIR model. And here is the reason why we choose to model this data. So when we're looking at the data on the state level, we notice that the number in the susceptible group is roughly the same as the state level population at any time t. So using this approximation, we can simplify this ordinary differential equation into this formula. And it is straightforward to get a solution of this ODE as shown in this equation. But our observed data are all counts. So we assume that the measurement error structure follows a Poisson distribution. In other words, we assume that under each time t, the observed state level actively infectious count follow this Poisson distribution. And the mean is parameterized in this way. So here capital N represents the state level population we adjusted for each state. And the alpha t parameter is associated with the time varying COVID-19 transmissibility. And the way that we choose to use n times alpha t as the mean of Poisson distribution actually reflects our assumption that this value should be equal to the right-hand side of the solution to IT. Here are more notations we defined for our change point detection. So in general, we assume that the change point should be in the collection of time points 1 through capital T, that is our observation times. And we introduce a binary vector to indicate the change point locations. So in this vector, each element is a binary value, so it is equal to 1 if the corresponding location is a change point, or 0 otherwise. And similarly, we can define a vector C. It is of length capital K. It will mark all the exact location of the change points. We can also define a segment label vector called Z, which is essentially just a reparameterization of our binary indicator vector gamma. And each element in this vector is defined as the cumulative sum of gamma vector until the current time t. And you will see this association with these 3 parameters in this example. So we assume that we know that these 3 points are the actual change points. So by definition, we can directly write down the formula of gamma and C in our model. And accordingly, we use the definition for Z to write down the corresponding Z vector. And by defining the segment label in this way, you can see the first 4 elements are in the first segment. and the 5th to the 7th element are in the second segment, and the last 3 are in the last segment. The change points, I'm sorry?
Sorry, 5 minute warning.
Okay. The change points not only separate the observations, but also our alpha parameter into distinct segments. Well, within each segment, we assumed that this following linear regression framework. So on the right-hand side, the XK is a segment-specific design matrix. So in this matrix, we can include information such as time or other explanatory variable that we believe is associated with the disease transmission. And the beta here is the segment-specific covariate effects, which is assuming that within each segment K, there is a unique association between alpha and the observed covariates. So after we have defined a distinct segment based on our change point, the next step will just be using a stochastic SR model to estimate the segment-specific reproduction number. Well, here let's see some results when we applied base miles to the real data. And we collected data from the Center for System Science and Engineering at Johns Hopkins University. And we consider 50 US states in total. And we make sure that the data collected for each state are in the same time range. Basically, when applied to the real data, base miles aims at helping us to answer the two questions, when and how. When is reflected by the change point location, how is reflected by the segment-specific reproduction number estimation. So here we see this figure again. So the change points are marked by the vertical lines, and the reproduction number is marked by the red dashed line. So in our analysis, we found that not only for Texas, but several other US states we analyzed, the change point detected for them matched pretty well with the times of publicly announced interventions. Another advantage of using a Bayesian method is to quantify our uncertainty in parameter estimation. So that's why you can see for each detected change point, there is a gray area representing the confidence interval. Similarly, for the reproduction number we estimated for each segment, there is a 95% credible interval associated with each of them. So here I would like to show several interesting downstream analysis. So first, we summarize the pattern of the frequently selected change point based on the result of 50 US states we analyzed. So the whole time range is divided into 3 categories. And by matching the detected change points for each state with this pattern, we're able to define a cluster label for each state. So for example, if we have a state and it has at least one change point in this first time range, we will mark the first element in its cluster label as change. Or if it does not have any change point in the first time range, we will mark it as stable. And we did the same thing for the second and the third time ranges. So for example, if a state is marked as change-stable-change, it means that this state does not have any change point in the second time range. So we can color the 50 US states we analyzed here based on the cluster label as shown in the bottom. So we can see from this figure, there is some very interesting spatial pattern. And our model is also able to make predictions. So here we compare the prediction of the new daily confirmed cases given by our method and the full SIR model I mentioned at the beginning of my presentation. So we quantify the prediction accuracy using the mean absolute percentage error. And as shown in the table in the bottom, we can see that the error provided by our model is much smaller than the full data SIR model. So to wrap up, our base models will use the change point information to help us understand the disease dynamics. When the data are partitioned into different segments, we're really trying to mimicking the disease dynamics under the changing conditions. And our Bayesian method can also provide provide uncertainty estimation in our model parameters. And in our regression framework, we're able to incorporate different types of explanatory variable to adjust for their effects. Well, from a practical point of view, we see that the change points we selected have some practical meaning because they matched pretty well with those publicly announced intervention times. And our state level clustering based on the change point pattern also revealed interesting spatial pattern. And last but not least, we see that incorporating the change point information could help us to get a better short-term forecast. I think I'm out of time, but here I listed the website that you can check out for our full model results. And I would like to thank my collaborator and advisors in this project. And thank you all for your attention. And here are my references. Can I take any questions?
I do not see any additional questions in the chat. Thank you all very much. This was a really interesting session, and I appreciate. Oh, is there one question here maybe? Let's see. Looks like there is one question if you're able to field it. In comparing change point timing to policy changes, how did you consider the lag between infection and system onset, incubation, and the lag between the system onset and testing confirmation for COVID?
Okay, I think that's a very good question. Due to the time limit, I didn't mention that our change point detection actually is very flexible. We didn't put any constraint on the exact time of those change points. We just let the data to speak for itself. Maybe there is a lag between the symptom or the testing, but our change point detection will just be based on the observed data. Yeah, so we are not forcing like the someday to be a change point just due to the intervention time.
Great, thank you very much.
Thank you.